Hi, my name is Marc-André Renaud, and I'm a researcher at McGill University in the Medical Physics Unit. In this presentation, I will highlight the key concepts of the RadCalc-MC module. This work is the result of over 20 years of expertise in applications of Monte Carlo simulations and radiation therapy. In addition to me, the people involved in this project are my colleagues Jan Sunjens, the director of the Medical Physics program at McGill University, and François Deblois, chief of the Medical Physics Group at the University of Montreal Hospital Center. With this project, our goal was to bring fast Monte Carlo treatment plan quality assurance to the clinic in a user-friendly way. Monte Carlo simulations are a statistical computational technique allowing us to calculate dose distributions in arbitrary geometries with unparalleled accuracy. It's only limited by our understanding of the basic laws of radiation physics. The way in which electrons, photons, and protons interact and deposit energy can be faithfully simulated using this technique. When doing these types of calculations, aside from knowing the interactions involved, one has to formulate a detailed description of the geometry and materials involved in the treatment unit, along with an accurate representation of the patient, typically from a computed tomography scan. The images on this slide show generic treatment head geometries involved in the production of megavoltage radiotherapy beams, along with a sample of particle tracks created using the Monte Carlo method. For conventional radiotherapy, these geometric elements typically include the target, the primary collimator, the flattening filter, and all the secondary collimation devices such as jaws, MLCs, applicators, and cutouts. Finally, there's also some physics parameters that need to be specified, such as the energy of the incident electron beam and the spot size. When people talk about Monte Carlo calculations in a clinical environment, there's always a perception that these methods are too slow to be useful. In practice, there are very powerful so-called variance reduction techniques that can be used to reduce calculation times. Variance reduction techniques are basically statistical tricks that make calculations more efficient without affecting the accuracy of the method. One of the major techniques that we used is called directional Bremsstrahlung splitting, and that basically involves only transporting photons that will actually contribute to the useful radiation field. Now at this point, there are many more variance reduction techniques that I could explain, and as an academic I would love nothing more than to devote the rest of this presentation to the wonderful world of variance reduction. But I have a feeling I'd have very few viewers left at the end, so I'll move on. The other main factor in speeding up Monte Carlo simulations is that it is cheaper than ever to buy multi-core workstations. And with Monte Carlo, simulation speedups scale almost linearly with number of cores. Twice the cores, half the simulation time. Speaking of simulation times, we've done some benchmarks using a 36-core Intel Xeon workstation. Most 6MV plans can be calculated to 1% uncertainty in between 3 to 10 minutes with voxel sizes of 2.5 millimeters. The exceptions are large irradiated volumes like chest walls or large prostate fields with nodal involvement. Those can take about 10 to 20 minutes to achieve 1% uncertainty, but since the targets are large, the voxel size can be increased to shorten simulation times back to under 10 minutes. A key factor to understand at this point is that treatment techniques have very little bearing on simulation time. For example, a 2-arg VMAT plan does not necessarily take longer to calculate compared to a single arg VMAT plan, or an IMRT plan. What matters the most is the target size. And the figures on this slide clarify what I mean by a typical small case and a typical large case. On the left is a small case, on the right is a large target. The last decade has seen a push towards more complex treatments with highly modulated fields and higher doses per fraction. There's also always a pressure to reduce margins as much as possible to spare healthy tissues. Now, all these things combined require more quality assurance to ensure patient safety, and they also put more pressure on quality assurance software. These cases also present themselves with a level of complexity that simplified dose calculation models cannot handle accurately. One cancer site I'm specifically thinking about is lung cancer treated with stereotactic radiotherapy, where small fields in highly heterogeneous environments very frequently occur. One of the well-documented areas where analytical dose calculation algorithms lose accuracy is at interfaces between high and low density areas, such as tissue and lung. In this figure, we show a dose difference color wash for a multiple beam IMRT plan, originally calculated using the Eclipse AAA algorithm, and then recalculated with Monte Carlo. In addition to small differences in the penumbra regions of each beam, we can see that AAA dose being hotter than in Monte Carlo immediately after entering the lung. Around the target, there are also 2 to 6% differences in dose due to the highly heterogeneous environment, a small mass of tissue surrounded by lung adjacent to the ribs. The typical sites where Monte Carlo is likely to show differences with conventional algorithms include lung SBRT, complex lymphoma cases near air pockets, esophagus cancers, and in general superficial tumors where many analytical algorithms are known to miscalculate the dose. Another area of complexity that can easily be handled with Monte Carlo is the MR-Linac, where the particle tracks in the Monte Carlo system can be easily calculated to take into account the presence of magnetic fields. Monte Carlo is widely considered to be the gold standard in terms of dosimetric accuracy, and our goal is to build confidence in the Monte Carlo models by showing customers great agreement in typical reference conditions during the commissioning process. Using our automatic beam commissioning, we will use measurement data that you've already obtained in a conventional Linac commissioning process to produce a customized beam model. Once this beam model has been created for your Linac, the software is ready to go for Plan QA, and that's it. No extra measurements or processing is necessary. In this presentation, I've highlighted how we use Monte Carlo for Plan QA in RadCalc MC. One of the key features of Plan QA software is that the results should inspire confidence. When there are disagreements between treatment planning systems and QA software, a rationalization process has to be followed to arrive at a suitable clinical decision. In the end, an inaccurate QA software may raise more questions than it answers. In RadCalc MC, you will have the definitive answer as to what the correct dose is in a particular clinical situation. Thank you for your attention.